So we'll go with the next presentation. Thanks for your time. And the next presentation will be Dr. Mark Treherne, Talisman Therapeutics Limited, UK. And the presentation title is Pluripotent Stem Cells as Models of Neurodegenerative Diseases to Support Drug Discovery Programs. Mark, can you just enable your video and just maximize your presentations into presentation mode?
Can you okay?
Yeah, it's fine.
Hopefully you can see the presentation too.
Yes, thank you. The stage is yours.
Let me just, it's just I need to remove, I just got your, everyone's video on the front of the thing. I just need to change, oh, yeah, right, hang on, just bear with me. So I'm from Talisman Therapeutics, and thank you very much for the opportunity to speak here today. My name is Mark Treherne, and I'm chairman of Talisman. We're a small company based on the Babraham Research Campus in Cambridge, UK. And we've been modeling neurodegeneration for a number of years now using in vitro models, which we think is a good system to support drug discovery in the neurodegenerative disease area. Some of you may know this already, but just as some background, this is how human stem cell models of neurodegenerative disease are produced. And we believe this is a very useful system for not only validating new targets in drug discovery, but also looking at pathway discovery and supporting screening activities. So we start with an induced pluripotent stem cell, or an iPSC, and these can be taken down different differentiation pathways by the techniques we have in the lab that are based around the original work of John Gurdon and Yamanaka. And you can take iPSCs and either make what we call neural rosettes. That means they have a neuronal or glial phenotype, or down an alternative pathway, which you can see here, and hopefully you can see my screen here. Here's the neural rosettes, and here's the other pathway that leads to microglia, or the resident inflammatory cells in the brain. So in some ways, they're equivalent to macrophages in the periphery. The neural rosettes can be induced to make neurons, and the neurons can be of various sorts. Often, for example, they're glutamatergic, the major excitatory neurotransmitter in the brain, or can be, for example, GABAergic interneurons, which sometimes have the opposite effect, or can be motor neurons, which reach out into the periphery and connect with, say, muscle. Or they can be turned into astrocytes, and often the astrocytes and the neurons have to be co-cultures for them to survive. And so we produce neuronal astrocyte co-culture models, which don't have any microglia in them, or we can produce the microglia and then combine those with the astrocytic and neuronal cultures. So what this enables us to do is to have neuronal cultures that have no microglia present, or neuronal cultures where microglia can be added to see what inflammatory responses can be studied in these systems. So we believe these are disease-relevant model systems because they're derived from patients that have some form of disease. We believe we've developed robust experimental endpoints, so we can actually develop these as screens rather than just research tools. And this enables a mechanistic understanding of the disease because the genetics carry through from the original donor. Many of those genetic characteristics that underlie the pathology can be recapitulated or found in the dish or the culture system. This enables us to be what we call decision-making data. So those are data which enable us to make early decisions about compounds. Do they work? Do they generate data that is consistent with the therapeutic hypothesis that's being generated or tested? And do they suggest that this compound or antibody or other therapeutic modality can be taken forward? Just some examples here of how we can apply these stem cell models to neurodegenerative disease, as the last slide was more about how you make them. So stem cell models, as we can see here, have cell lines and genome engineering. So what you can do is you can induce or detect genetic mutations. So the genetic mutations may be there originally from the donor, or you can use other techniques to induce them. And you can also run, for drug discovery purposes, phenotypic or genetic screens. So you can say it's a genetic form of a rare form of Alzheimer's disease, for example, or you can just look at the phenotype and say this phenotype represents the pathology that's often seen. And here are some pictures of those cultures. So just want to give you some examples of some of those pathologies, and we just showed you a snapshot of the pictures in the previous slides. And what we have here, and we mentioned sort of rare forms of disease or very strongly genetically driven forms of Alzheimer's disease or familial Alzheimer's disease, by which we mean they're carried through families over multiple generations. And here are some of the publications here that underlie the research we've shown today. And obviously a well-known mutation is the presenilin-1 mutation. It's autosomal dominant and has been shown in many labs, including our own. This is a causal mutation from familial Alzheimer's disease, which is thankfully quite a rare form of the disease. Another example here is the APP or amyloid precursor protein, what's called the London mutation, which is where it was originally discovered. And again, this is an autosomal dominant cause of mutation, and this has been very well characterized in our labs and other labs as well. Other forms of disease are frontotemporal dementia, and this is associated less with amyloid, but more with microtubule associated protein tau. So this is more of a tauopathy, if you want to use that term. And again, is another autosomal dominant causal mutation for frontotemporal dementia. And other sorts of things we can look at, for example, are triggered receptor expressed on myeloid cells. For example, TREM2 is shown here, and these are known in the labs, our labs and other labs, to increase the risk of both frontotemporal dementia and more conventional Alzheimer's disease. Another model which you think is really quite interesting is a trisomy 21, which is Down syndrome, or TS21, where there is an extra copy of chromosome 21, which overexpresses certain forms of disease. And here's a publication from our lab, Shi et al. From 2012. And in trisomy 21 or Down syndrome, there is a high incidence of Alzheimer's and dementia. And we and others believe this is because some of the genes on chromosome 21 are overexpressed. And often this is seen by seeing abnormal forms or overexpression of both amyloid and the tau protein, which are thought to underlie various forms of dementia. So here's some pictures of human iPSC derived neurons that are derived from healthy controls as well as from patients who have the condition. And we've developed in the lab sort of proprietary methods based on published protocols to reliably regenerate projection neurons and neuronal astrocyte cultures that I showed before. And you can see that we use nanostring technology. So this allows us to quality control or QCs the progenitor cells to ensure we have a consistent and successful generation of mature neurons, because we want these neurons to be as mature as they possibly can to mimic the human disease rather than be very early embryonic-like neurons. In vitro differentiation mimics the human in vivo neurogenesis, so we can believe that the neurogenesis we see in our culture systems is similar to that that's seen in vivo in humans. And the cerebral cortex complexity is recapitulated. We see structures that you can see on the right here showing some of the complexity that's seen in, say, the human cerebral cortex. And network activity, so this is electrical signaling that we believe is relevant to demonstrate maturity of the synaptic connections, can be seen around 60 days or more in culture. And the cortical interneurons are generated from neuroepithelial precursors. And we've got these cultures working in anything from 6-well plates up to 384-well plates, which are obviously more useful for higher density formats in screening, supporting drug discovery, either medicinal chemistry optimization or early screenings of medium-sized libraries up to 20,000 compounds. Here are just some pictures that we're showing here, characterizing these cultures with beta-3-tubulin, which is relevant to the neuronal phenotype, and GFAP, or glial fibrillary acidic protein, which is more relevant to the astrocytic cell type. And these data are really just showing some pictures to demonstrate that what these cultures look like, having explained why they're important here on the left-hand side of the screen. As I mentioned earlier, these iPSC neurons form active networks. They're electrophysiologically active. And here's some pictures of these networks, these culture systems, through phase contrast and also through Neurolight, where you can see some of the connections and synaptic connections that are produced. And you can see here that we show intensity levels, and we can see waves of neuronal activity, as you can see in these spike patterns here over 60 seconds, a minute or so. I'm just going to try and show a video of that, and hopefully that will come through, but bear with me while I start the video. And you can see that these neurons are firing in bursts, and that the intensity of those bursts are shown in the slide here over a period of 60 seconds or so, a minute or so. So we believe this is evidence that the neurons have synaptic connections and are firing regularly in synchronized bursts. So I mentioned before the astrocytes, the glial cells that are there to provide support to the neurons, and actually very important in maintaining the health and the recapitulating in our hands the pathology of some of the diseases that we're interested in. And cortical astrocytes follow a similar developmental pathway to cortical neurons, because during development both are required for normal development of the brain. And they progress from relatively immature neurons to immature astrocytes expressing GFAP, glial fibrillary GFAP, a protein that identifies the astrocytes. And eventually, in long-term culture, they have the phenotype of mature astrocytes expressing other markers, such as fermentin and others that are markers for maturity seen in the more adult brain. So here's a picture of some of those cultures at day 35, going on to day 57, and you can see they look quite different at day 57. They're starting to become mature now, and you can see that if you use different stains, you can see different patterns. And by over 100 days, they're actually looking really very, in our view, quite mature, and therefore much more closer to something that you would see in an adult human brain. So the issue here is there's a very long differentiation and maturation to get these. It's more than 100 days in culture, because if you work with early cultures, you're looking at astrocytes with a relatively early or immature phenotype that we mentioned earlier. As I said up front, we're also very, very interested in the role of microglia, the resident inflammatory cells in the brain. And microglia are produced differently, as I explained earlier, but also are very interesting drug targets and important cells to use in supporting drug discovery, particularly if you're trying to treat neuroinflammation, for example. So showing a bit of differentiation into forming the microglia. So the iPSCs are directly differentiated into microglial cells via hemopoietic precursors. So these come from a blood origin in the blood. They have a characteristic morphology and protein expression of in vitro microglia. And here is showing you some pictures of what they look like and the staining that we've done of these cultures that make us believe that these have the characteristics of microglia and not other cell types. So you can see here they start from pluripotent stem cells. They then form embryoid bodies. And from day forwards, you get primitive macrophage-like precursors. So macrophages in the periphery have many similarities to microglia in the central nervous system. And then you can label these up with markers such as CD45 and TREM2 that I've spoken to earlier, which are deemed to be important in many of the pathologies seen in microglia or related to neuroinflammatory diseases. And here's the publication of these data from 2018. So what I want to talk about now is a bit that microglia express characteristic proteins and robustly respond to inflammatory signals such as LPS, lipopolysaccharide, that you would expect these microglia to do in vivo, and in fact they do something very similar in vitro in our cultures. So these are 15-day microglia monocultures. So it's just microglia. There's no other, no neurons there or astrocytes at this stage. And you can see that they light up with IBA1 and CD68. And if you put the two things, you've merged these two images, you can see they're co-located in the same cells as shown by the marker here. And we believe that this is relevant relevant to demonstrating that they are genuinely microglial-like and not something else. And you can see the relative size of the cells here by the mark here, which is about 50 microns. And the point here again is that the morphology you see is very classically like a microglial morphology that you would see in vivo. So this allows us to quantify those markers, to look at the microglial number and density, the size of the microglia, and any morphological changes, because as they're stimulated, the morphology of these cells often changes. And here's some data here where we're looking at the effects of TNF alpha release. And you can see that you get a very strong response to TNF alpha and other sort of pro-inflammatory cytokines at, say, day 15 in monoculture. So to see this sort of morphology, you don't have to culture them so long as you do with the neurons, which to get maturity may take you up to 100 days in culture. You can generate usable microglia much more quickly, which means in some ways they're easier to use in early stage drug discovery because there's less culturing required to get them to that point. The other thing I want to say is that iPSC microglia show evidence of inflammasome activation. So this is sort of, we're saying as markers of inflammation. They've been triggered to respond to an inflammatory stimulus. And the point is they're highly phagocytic, which is part of their key role in the brain. So here we can see, this is a phase contrast of some microglia. And that when you add lipopolysaccharide and other inflammatory cytokines, you can see their morphology changes quite markedly. They start to produce these processes, which is very characteristic of them being stimulated. So here's some data here taken from images such as those on the left. And what we're looking here is IL-1 beta release, so interleukin-1 beta release. And you can see, you can look at primed and unprimed, and you can see that with ATP here, you see a massive, in fact, it's such a big response, we've had to sort of cut the graph here. So they very much respond in producing IL-1 beta, which is a key marker of microglial activation. And also you can see in the far right here that you see phagocytosis, that when they're activated, they gobble up a debris, again, which is part of their function that's well known and well characterized in the brain. So we can see many of the characteristics of in vivo microglia in our culture systems. As I mentioned earlier, we're very interested in co-culture systems. So this is looking at the microglia when they're added back with the neurons. So they're not just on their own now, they're interacting with the neurons, which is what happens in vivo, of course. And we've been looking here with 2-photon imaging. That's looking over a 2-hour course. And here you can see that these neurons have been cultured for about 10 days. Here we're using an opera system. It's an opera phoenix system that allows us to image these. And what this allows us to do is to demonstrate that they can be co-cultured for up to, in this case, 49 days. And again, the images that are seen are highly characteristic. So I'll just show you those two videos now. So this is at 8 days. And you can see they're really quite activated here. You can see the philopodia going out there and reaching out into the environment. And if you go to 49 days here, you can see another video where they're really clustering in onto the site that where the phagocytosis is going. So they're clustering into the site where they need to do what they do in the brain. But we've recreated this in vitro in a dish. And the red here is showing where the stimulus is, and that is what is tracting them in to that site. So moving on, just want to sort of summarize where we've got to so far before I go into a couple of case studies. I think I have a few minutes left for this presentation. So I think we've got a lot of expertise and experience in molecular and cellular biology and neurodegeneration. We developed human-induced pluripotent stem cells, and we use these to develop disease models for high-throughput imaging, screening, as well as target validation. So we think these are really good tools to get to decision points, pivotal decision points early in the drug discovery process before you need to go towards animal models. Our models are iPSC-derived cortical neurons, interneurons, and astrocytes, which we believe recapitulate the complexity and pathology that's seen in vivo, seen in humans. And we've enabled multiple assay readouts. So depending on what you're interested in drug discovery, we can look at imaging, protein quantification, high content imaging, and neuronal activity through electrophysiology or using other markers of neuronal excitability. The human iPSC-derived microglia express characteristic proteins. These are proteins that we know are very relevant and unique to microglia. They respond to pro-inflammatory stimuli such as LPS. They exhibit a robust inflammasome activation, so they release the things like IL-1 beta that we showed earlier, and are highly phagocytic and act as microglia are expected to do in co-culture systems. So that's a little bit of background to the overall technology platform. Now I just want to go through a couple of case studies, or how much time I got, maybe one, just to talk about how these can be used in drug discovery. So this is an analysis of mitophagy, where it's thought that the mitochondria have a dysfunction, and the neurons are trying to clear the dysfunctional mitochondria so that they can be replaced with healthy mitochondria. So mitophagy is clearing the abnormal or damaged mitochondria. So what we're trying to do is to show how efficacy of novel compounds, which we've just shown here as examples A and B, to enhance mitophagy, and these are in rodent models. And in this particular thing, we looked at a human neuronal model to enable basal and induced mitophagy to be quantified. So here we use this marker TOM2. So we were looking actually at the clearance of these mitochondria in this particular system. And we use these data to assist decision making and project progression in here, because the interest was really to understand how these drugs could accelerate mitophagy, could increase the clearance of the damaged mitochondria, with the idea that those damaged mitochondria would then be replaced with more healthy mitochondria produced in the cell body. So here they were looking at trisomy 21, which is the Down syndrome neurons I mentioned earlier at 6 days, with DMSO, that's the vehicle control, and baflomycin, and then the test compounds. And we saw mitochondrial redistribution following application of the compound of interest in this particular example. So what does the data look like when you take the images and analyze it and to put it in graphs? I won't go through all of the details here. But really we're looking at measuring enhanced mitophagy in the presence of a novel compound, which is at this stage it's confidential, so we're just calling it A. And here we're looking at co-localized TOM20 and LC3. And what you can see with these various different pharmacological manipulations, you're looking here at the mitochondrial area. So the number of mitochondria there, and you can see that in this particular example, the mitochondria are cleared. So what it's showing is it's clearing the damaged mitochondria, which is the sort of therapeutic effect that's clearly desired. This is another case study, a different project, again relevant to drug discovery, where mitochondrial express characteristic proteins and are functional in the co-culture, some of which I've shown you earlier. This was a project that was funded actually by Innovate UK, the UK's innovation agency, and they funded the development of this model. So thank you to them for those funding. And here you can see that in this model, clearly this is in the neurons, you're seeing tau. You're seeing tau, the protein referred to earlier. We're seeing other IBA1 here imaging. And you see when you can merge the two, you can put the two images together, and you can look at the co-localization of these different markers. Again, a similar thing, we're looking at this marker here, NEUN and CD68. You can image them separately and then put them together to see, look at co-localization to see, how the microglia and the neurons are interacting in these co-cultures. Here's some data here, also looking at TNF-alpha or cytokine release. And here we're looking at vehicle control, LPS, and other inflammatory stimuli. And you can see that again in these cultures, they respond to these stimuli in the way that you would expect from a pathological perspective. I think I'm beginning to run out of time now. I don't know how much time do I have left. Is it maybe a couple of minutes?
Yes, yeah, you have a couple of minutes, not an issue.
So I'll just sort of finish with this last slide because I've got some other case studies we could show. And here in this particular example, we are looking at the percentage of LPS and the response to these in terms of TNF-alpha. So you can see we get a big response, but when you put these inhibitors in, you inhibit the response. So here we're looking at actually an old Pfizer compound here, CP863187, and you can see you get a dose response curve here. So it's a relatively potent compound, whereas you can see that this other compound here is producing a less response. So you can see that by inhibiting sort of pathways such as PDE4s or P38, so these are phosphodiesterase and kinase, you can actually inhibit these responses. And we can show that we can recapitulate this pharmacology in these culture systems that other people have seen in vivo and elsewhere. So LPS and other inflammatory cytokines produced a robust TNF response in microglia neuronal cultures. The cultures enable us to generate pharmacology, and you can see that it's pretty much what other people have reported, that the Pfizer compound here has an IC50 of about 10 nanomolar, whereas this compound here is significantly almost sort of 2 micromolar. So you're beginning to see the pharmacology that have been reported. So we know these models are going to be useful for testing other compounds with novel mechanisms, as well as compounds with known mechanisms in such culture systems. So I'll just finish here. We also look at model validation with altered microtubule dynamics in frontotemporal dementia that I referred to earlier. And we can compare in these examples non-disease control with MAPT mutations. I'll just show you what happens in the non-disease control. And you can see here that we're looking at microtubule dynamics. That's what's in the normal neurons. But if you then move to the MATP purines, you can see that you see really quite a different dynamics in these videos. So we're showing live image of abnormal microtubule dynamics in the cells because this is very important and particularly relevant to frontotemporal dementia where there are well-known tau mutations, and allows the exploitation of microtubule biology that led to the identification of a potential new target. So not only can the culture systems that I referred to earlier be very useful for looking at screening with known targets, they can also be very useful in terms of looking at novel targets and target discovery. So we've looked at various things: kinase inhibition, phosphodiesterase inhibition, effects of tau and amyloid, and we think these are great models to support many forms of drug discovery when people are interested in treating Alzheimer's disease and dementia more generally. But thank you very much for listening. I will stop now and very happy to take any questions if anyone has any.
Yes, thank you, Dr. Mark. So is there any questions from audience? Anybody? Okay. That's fine. Thank you, Dr. Mark. And it's very glad that we have your presentation at this webinar. And with this, we will end up the session for the day. And we'll be having our day 2 tomorrow at 11 o'clock in the morning GMT. Hope everybody will join with the same link. Thanks for your time. Have a great day.
Thank you very much for sharing.
Bye-bye.
Much appreciated.
Bye-bye.